Hello everyone! Today we're going to be talking about the microbiology of beer making. Beer is an alcoholic fermented grain beverage that has an important presence in human history. Beer has played an immense social and religious role. When sanitation practices were scarce, beer was often preferred over water, as its alcoholic content made it safer to drink. Beer consists of 5 main ingredients: water, grains, hops, yeast, and adjuvants like fruits and spices. Water is the base ingredient that provides the bulk volume of beer. Barley is the most common grain used to produce malt, a cooked-down form of the grain, which provides the sugars to feed the fermentation as well as the primary color, taste, and texture characteristics of the beer, including the foamy head. Hops are added to give beer its characteristic bitterness and help stabilize beer foam. They also have antimicrobial properties that limit the growth of undesirable spoilage bacteria. Yeast is the single-cell organism responsible for the transformation of sugars into ethanol and carbon dioxide during the alcoholic fermentation period. There are 2 types of yeast: top fermenting, Saccharomyces cerevisiae, and bottom fermenting yeast, Saccharomyces pastorianus, based on where the yeast settles once the fermentation is complete. Normally, Saccharomyces cerevisiae is the yeast of choice to make ale beers. Lastly, adjuvants are added to create a second layer of flavor and aroma profiles to complement the brewed product. Let's walk through the process of making beer. First, the grains are milled. This allows for the penetration of liquid inside the grain and facilitates the extraction of sugars. The ground grain is cooked in water at high temperatures for 2 to 4 hours to obtain wort, a liquid containing the sugars, flavors, and aromas of the barley grain. The grain solids are filtered out using a slow sieving process. The collected liquid is boiled again with the addition of hops in order to extract the enzymes, bitter compounds, and antimicrobial agents desired in the beer. In the fermentation tank, yeast is added to the wort and left to react for about a week. Yeast is just like Goldilocks. The addition of yeast should not be too much or too little. Too little leads to a slow initial fermentation, but too much will lead to a competition between yeast causing poor growth. The temperature of the wort should just be right. The wort must be first cooled to prevent killing of the yeast, but it should not be more than 5 degrees Celsius cooler than the yeast to prevent cold shock. Beer is subject to microbial spoilage producing off flavors. Beer is sterilized through pasteurization or cold filtration before being bottled to prevent possible growth of beer spoilers and also to extend the shelf life. Through various stages in the process, samples are taken through sample cocks present in the production line and tested in the microbiology lab for contaminants. And there you have your perfect pint. Cheers!